Friends, hello everyone. My name is Evgeny Snegiryov. I am the head of the aquix laboratory, and I have wonderful news for you today. For a month now, together with Omsk Agrarian University, we have been conducting research on our IZONEUM water. And now I suggest you to go to the university with us and see the intermediate results of our experiments. They are already promising. Let's go. My name is Tatiana Vladimirovna Boyko. I am the head of the Department of Diagnostics of Internal Non-Infectious Diseases, Pharmacology, Surgery and Obstetrics, Doctor of Veterinary Sciences, Associate Professor. These are all green today. Yesterday there were 3 and today they are all. And today it's already more difficult to catch the difference, only in length and in quantity. How many seeds with this length or another? We have already differentiated everything. In this case, a test object is presented. These are cress seeds and IZONEUM water, and its effect on the growth and development of the root system and supports were tested. What did we evaluate? We estimate the seed's germination rate and the length of the seedlings. It was recorded under the certain conditions. The water was tested in several variants. This is fresh water, water after storage for 5 days, and water that was sorted in frozen conditions also for 5 days. In addition to the fact that we place these seeds in cups and evaluate their growth and development according to the method, in the future we conducted research using a simple rule and measurement. These studies are already presented in the computer on the screen. The results indicate that the IZONEUM water affects the germination of seeds, and the number of fruits with a length of more than 5 cm is greater than in the control samples. This is clearly visible, and it only indicates that the activity is higher than when treating seeds with IZONEUM water, namely fresh. If you look at the number of fruits, which are also quite long, but the IZONEUM water was after freezing for 5 days, then they are less than fresh water, but more than distillate water. That is, the activity decreases, but it persists. It depends, but it persists because if we look at the control and count the number of fruits that are long, then there will be fewer of them than in this sample. And this is visible. And this is not one sample. More than 30 repetitions were carried out so that this pattern could be established. We also tasted the water after storage in the dark place and in the light. That is, in this case, number 27 is in a dark place, number 28 is in the light. For control, we had distillated water, and we also see clearly that the number of seeds sprouted and having a root length of more than 5 cm is greater in the experimental sample than in the control ones. So, what can we say? We can also say that the activity of this water is preserved of even expressed 5 days after storage in the light or in a dark place. There is activity. And a test system such as lettuce seeds, they caught this pattern. That is, all the studies were conducted under the same conditions. One temperature regime, one humidity. All the conditions were absolutely identical. And this gives us the right to testify that yes, indeed, for 5 days, the stimulating activity of water on seedlings is preserved. These results are presented on the fourth day. It is the measurements of the root itself that we will carry out on the fifth day. That is, we will evaluate the result on the fifth day, but it is already clear today, on the fourth day, that germination is quite active in all samples. These results were monitored every day, and many repetitions have already been carried out. More than 30 repetitions have already been carried out. A statistical analysis of the result obtained has been carried out, and we will now introduce you to this result in another room. Another goal of the experiments was to evaluate the effect of IZONEUM water on the growth and development of microscopic fungi. If we look at the cups that are in the kelp, we will see the first and second cups are distillate water, and there are colonies of various microscopic fungi on the discs, and penicilli and aspergillus and so on. The following cups are experimental. On these experimental cups, we also see that the discs were treated with IZONEUM water. We see that the growth of fungal colonies persists. However, if we look at cups 3, 4, and 5, 6, the difference is obvious. What is the obvious difference? Even a person who does not understand and does not know anything about mushrooms will still distinguish these green colonies in this cup and in this cup. He sees that there are more colonies in the fifth and sixth samples than there are in colonies in the third and fourth cups. It means that some factors hindered the development of penicillia precisely in these conditions. When activated water, freshwater IZONEUM was used. On the contrary, the sample of IZONEUM number 2 somehow stimulated the growth and development of fungi, namely penicillia. This is only the first repetition. We will make several more such repetitions in order to understand whether this trend or pattern persists, it is, or it is an accident. If earlier we did not observe the growth of the Fusarium colony, now it is already appearing. But what does this mean? Well, it is premature to draw conclusions. As long as we see one compared with distillate water, and these are only 2 samples of distillate water. If we compare them in this sequence, distillate water, IZONEUM active and IZONEUM sample 2, then of course colonies of 2 species are observed. But water number 2 activates the growth of penicillium colonies. That is, the number of penicillium colonies was higher. Whether this trend will continue or not, we will observe. Activated water in one case did not give the growth of penicilli, but for some reason distillate water also did not give. That is, we just need to collect material for statistics so that we can see a pattern. Well, so far we have received such results. In these cups, another result is presented. When the IZONEUM water was not tested in its pure form, that is, by wetting the filter paper discs and placing them on the nutrient medium, while the mushrooms were taken from the room in which the research was conducted. In this case, the IZONEUM water was used as an extractant. That is, we took hay as a sample, moistened the IZONEUM with water, fresh water and the IZONEUM sample number 2, and the wetted discs were placed on the nutrient medium. And what do we see? We see that the development on microscopic fungi is quite high in all samples. where we have distilled water. Here we have sample IZONEUM number 1 and sample IZONEUM 2. The number of colonies in all the cups is quite large. It was possible to count them only at an early stage. In this case, the culture is already the 6th day. We will continue to observe this culture, what will happen. Another test object for studying the influence of water was Stylonychia. Stylonychia are the simplest organisms, single-celled, which allow us to assess the influence of water by the following reaction. First of all, survival. They live or die, but also the next important point is how they divide, that is, how the water of the IZONEUM affects their reproduction. Here are two criteria by which we evaluate the influence of this water in general. We also tested water in different versions. It was both a storage option and fresh water and sample number 2, which is unknown to us, and we used this standard technique to determine the total toxicity. But later, that is, where we use such microplates for planting Stylonychia and their subsequent counting, but when we realized after the first experiment that the water is not first toxic, that is, the survival rate is 100%, we removed this component from our studies and began simply to reseed these protozoa from one sample of water to another, that is, every time we brought fresh water or every time we took water after storage, we transferred them from one cup to another cup. Also, more than 30 plantings have already been carried out, and we see a sufficiently high safety of these stylons. In addition, we found that fresh IZONEUM water increases the number of long forms of Stylonychia. Long forms of Stylonychia are those forms that are ready for division. That is, even without knowing what is containing in this water, this is a blind experiment for us, but we see that the division process of these single-celled protozoa are activated. These are results we received. We can look at this Stylonychia under a microscope and clearly see how they exist. Here they are running, but they are doing it quite slowly now. Now the admission temperature is, how much does we have? Oh, 15 degrees. The result of our research indicate that the IZONEUM water, which is fresh, actively affects the growth and development of both protozoa and plant objects. In addition, it should be noted that this unique feature persists for 5 days, even if not the extent that we saw on fresh water, but nevertheless, this activity is present. If we talk about the results that we see on mushrooms, it should be noticed that fresh IZONEUM water affects the growth and development of certain types of mushrooms. But these results are intermediate, and of course, a series of experiments will be conducted in order to understand which types of mushrooms grow more actively and which types significantly reduce their activity.